Thank you very much for inviting me to speak at this. We are always eager to be part of a community that tries to promote active learning. So if you just go to the next slide, and even the next, this is just a breaker. So here is just a quick summary of what we do. We are a department at Novo Nordisk, a large pharmaceutical company. We are within the research and development, 500 people working exclusively on digital science and innovation. And some of us are working on active learning, trying to bring that out into the labs and do, well, you were sort of burying DOE in your initial speak. We are even fighting just to get that into the labs. So in any way we can help the wet lab scientists better understand the data and do experiments smarter. And sometimes it's just giving them a design that's all they can solely really manage. And sometimes it's making very advanced autonomous systems, self-driving labs. But it is really all, if you look at the right, it's about the perfect recipes to help them move forward. And it's about the optimal learning per experiment. So if you go to the next slide, this is one of the tools we have to do that. We have a internally developed Bayesian optimization package called Process Optimizer. It is available on GitHub, totally open source, and it is by no means bleeding edge, very much on purpose in many ways, because what we are really focusing on is having our organization adapt these methods rather than being front runners on the technological level. And that leads to the right side, because some of you might have experience trying to get your colleagues who are less familiar with the programming to use these methods, is that it can be quite a battle. So we have gone through various iterations of what you could call change management, trying to have people use these for developing their assays or what else you might use it for. And one of the things that really worked was when we, surprise, surprise, made a very intuitive front end for using Process Optimizer. We did that in collaboration with a couple of research institutes in Denmark. We got some actually external funding for this. So for a long time, this was totally open source, paid for by a public grant to have access to this and also the nice front end. And we saw a lot of different companies from various industries, so glue making and carpets and whatever, using our Bayesian optimization package to improve their products, which was quite fun to see that a lot of these smaller companies were really eager to adopt this methodology and see what they could do with it. There are a lot of nice cases. So the figure on the right, that's how it looks when the research institute tries to sort of sell it now. If you go to the next slide, you'll see how it actually looks when you go in there. And it's not that different from it. So it is a way of specifying your parameters and making constraints. And then you can set up your experiment run. You can also get batch experiments one at a time, and it'll help you navigate your space in the most efficient way. This is just an example with baking brownies, hence the name Brownie Bee. One of the early examples to prove the value of this in a way everyone could understand. If you go to the next slide, you go into something slightly more high-tech than brownies, because we also do other things. When we have some users who are more tech-savvy that we can work with, we can do more things. This is just a quick figure showing how we made a chromatographic method scout, which is closed-loop HPLC method development. So we have our Bayesian optimizer under the hood here, optimizing the parameters for HPLC methods in an autonomous way, able to run through the weekend, which is really a cool thing for us. We have 700 instruments doing this, producing more than a million chromatograms every year. So you're really talking volume here. And I think that brings to my final slide. I was asked to bring a question, and as I said, we are really focused on adoption. How can we make our organization actually use this? So I would like to discuss how we as scientists working on this methodology can work to get it to use really and generate impact in the organizations we're in.
Wonderful.